Hello, my name is Priscilla Sano, and I'm with Grand Canyon University. Today, I will be presenting my topic on ion channels and neuronal communication for my biochemistry class under Professor John Wool. In today's objectives, I will cover an introduction into the topic, cell membrane charge imbalance, voltage-gated ion channel overview, voltage-gated sodium channels, voltage-gated potassium channels, voltage-gated calcium channels, gated ion channel responsibilities, neuronal action potential explained, the propagation of action potential, the cease of neurotransmission, and some interesting examples of toxin effects on ion channels. Within the body, there are obviously different types of cells. There are a class of cells that are called excitable cells. A couple of examples of excitable cells are going to be neurons and muscle cells. These excitable cells in the body change an external signal into an electrical signal, and this is responsible for nerve impulses, muscles contracting, hormones, sensory processes, learning, and memory. These impulses can only come about due to ion channels. Ion channels are responsible for moving ions such as sodium and potassium across a cell's membrane in response to any external stimulus. Ion channels are different from pumps in that they are in continuous need of large amounts of ATP and other molecules to fuel them. Gated ion channels open and close if its receptor has come into contact with a ligand, a neurotransmitter such as acetylcholine, or a change in potential. The sodium and potassium ion ATPase imparts a charge imbalance over the cell membrane. This occurs because there will always be 3 sodium ions carried out of the cell for every 2 potassium ions taken into the cell. The inside should be more negative with higher levels of potassium, and the outside should be more positive with more sodium ions. If sodium ions are to open, the ions will travel inside, causing depolarization to occur. If potassium ion channels open, the ions will flow outward, causing hyperpolarization. This will cause subsequent ion channels to open in order to carry an electrical impulse down a neuron and across a synapse that will cause the release of neurotransmitters. The depolarization or repolarization of a small area and the spread into the next channel is a passive process, and that's called either a cable spread or an electronic spread. The outside of the membrane and the inside of the membrane are better conductors than the membrane itself. These gated ion channels are responsible for transmitting signals by 2 methods. 1, they can fluctuate the cytoplasmic levels of calcium ions, and 2, they may change the electrical potential. A neuron will experience both methods mentioned in order to transfer electrical signals among neurons or from a neuron to a myocyte. Neurons carry action potentials from the body of the cell to the axon. At that point, the electrical signal influences the release of a neurotransmitter at the synapse, and that will carry the signal to the next destination. There are 3 types of voltage-gated ion channels based on ions. There are sodium voltage-gated ion channels, potassium voltage-gated ion channels, and calcium voltage-gated ion channels. For this slide, we'll focus on voltage-gated sodium channels. Along the cell body of the neuron, which is in this region, and throughout the axon down into the terminal, we are going to find voltage-gated sodium channels. When at rest at an electrical potential of anywhere from minus 60 to minus 70 millivolts, the channels are going to be closed. Okay, and we see that down here in this graph. Now, if depolarization takes place, the channels are going to open in response to a neurotransmitter such as acetylcholine. And this is where sodium ions will end up traversing the membrane and going inside. And that will cause the inside of the membrane to become more positive. And with that, we see a spike in this graph representing depolarization. There will also be voltage-gated potassium channels along the cell body and throughout the axon and close to the synaptic terminal. When the voltage-gated sodium channels open, the voltage-gated potassium channels will soon follow and they will open as well. When the sodium ions move in, causing more depolarization, there will be a hyperpolarization that takes place as the potassium ions are going to move out. And we see that within this graph. So as sodium ions were influxing, we saw a spike causing it to become more positive. But as your potassium ions are moving out, it's causing a repolarization and it's starting to become more negative. Voltage-gated calcium channels are concentrated in the nerve terminals, so down at the end of the neuron itself. They are going to open when depolarization and repolarization occurs, and that triggers the release of acetylcholine down into the synapse. So once we do have acetylcholine present in the synapse, it can signal another neuron or the subsequent muscle fiber. Neuronal action potential explained. The plasma membrane is going to be polarized, which means that it is negative internally. A neurotransmitter or a change in potential causes voltage-gated sodium channels to open, and we see that right here. Sodium ions are now going to flux into the membrane, ending in depolarization of this specific region. This area being depolarized will cause the next voltage-gated sodium channels to open, and then sodium ions will rush in, and the signal gets carried along the body of the neuron in one direction. There's a refractory period that occurs after the channels open to ensure that the signal travels in only one direction. Once depolarization is over, the membrane needs to get back to resting potential, so sodium channels close, and they're not going to open during this refractory period. This ensures that another action potential can't take place until repolarization is complete. Voltage-gated potassium channels are going to open slightly after the depolarization takes place, once the sodium channels open. So as sodium is rushing in to the cell, causing the depolarization, their channels will close, and then potassium will end up rushing out to repolarize the membrane locally. So this repolarization of the membrane locally ensures that the inside, once again, will become more negative, and that will allow the action potential to move along the body of the neuron. Potassium moving out will hyperpolarize the cell, and the membrane potential will be more negative than at normal resting potential. But with that, voltage-gated potassium channels will close and potassium leakage channels are still present. So some of the potassium will leak out, bringing the hyperpolarization back to normal resting potential. Sodium channels at this point will be ready to open again. So when the wave of depolarization and repolarization travels down the cell body and migrates towards the axon, voltage-gated channels made of calcium are going to open, and that allows calcium ions to influx. The high concentration of calcium ions inside of the membrane induces the synaptic vesicles to merge with the presynaptic membrane, which is right in this region. The vesicles possess neurotransmitter molecules called acetylcholine, and they're going to be released into the synaptic cleft. Acetylcholine will then connect to the postsynaptic neuron receptors. Ligand-gated ion channels open, and they're going to allow the influx of calcium ions and sodium ions to traverse into the postsynaptic cell, and that depolarizes it, and it's called an excitatory postsynaptic potential, or an EPSP. So from this point on, the same cascade of events will occur as previously discussed to carry an electrical signal throughout this neuron. The propagation of the action potential is made possible by two things within the axon, and we call them the myelin sheaths and the nodes of Ranvier. The myelin sheath is going to be an insulated area of the neuron, and the nodes of Ranvier are not insulated, and these will possess sodium and potassium voltage-gated ion channels. So an action potential has to be able to transmit a signal to another neuron, and therefore has to travel the entire length of the neuron. Now, the speed is affected by the resistance to leakage and how wide the axon is. So myelin is going to insulate the axon, and that current is not able to leave it. In cases of certain diseases like multiple sclerosis, it's known for demyelination, and that causes an action potential to slow down because those areas will now allow leakage since they are no longer insulated. Within the nodes of Ranvier, these are areas in the axon that don't contain the myelin. These areas have voltage-gated sodium and potassium channels. Sodium and potassium ions moving through these channels help to regenerate the action potential, and it's called saltatory conduction. Without the nodes of Ranvier, the entire axon would actually need the sodium and potassium channels. And that would slow the action potential down, and they also help to conserve energy, because wherever we have any type of voltage-gated ion channels present, they're going to require large amounts of ATP to be present. The cease of neurotransmission is brought about by three factors, and we are going to discuss diffusion, degradation, and reuptake. The end of the signal transmission will entail that the neurotransmitter will need to be removed from the synaptic cleft. So this will ensure that everything returns to a resting state and that the neuron will be ready for the next signal. This will happen when the acetylcholine will end up diffusing away from the synapse naturally. It can be degraded by enzyme activity, or reuptake could actually occur by the presynaptic neuron. When conducting my research, I found a topic that I thought was interesting, so I thought I would implement that into my presentation. And I found examples of certain toxins and how they affect our ion channels and how that can be detrimental to human health. So within that, we'll be talking about dendrotoxin, tetrodotoxin, batrachotoxin, cobratoxin, and saxitoxin. Now, the black mamba snake makes a toxin that's called dendrotoxin. That blocks the action of voltage-gated potassium channels. The pufferfish produces tetrodotoxin that affects voltage-gated sodium channels. It does so by forming a plug in the pore and blocks the pore so that the pore will no longer allow sodium channels to influx into the membrane. Batrachotoxin is found in poisonous frog skin, and it causes activated voltage-gated sodium channels to stay active, causing a huge depolarization. That will block the transmission of nerve signals. Certain cobras make cobratoxin. Cobratoxin does not allow the acetylcholine receptor ion channels to function properly. Cyanobacteria in freshwater and dinoflagellates in marine water make saxitoxin. Now, these are small organisms, so usually you have larger organisms that feed on them. We have filter feeders and fish. So as these filter feeders and fish are eating large amounts of cyanobacteria and dinoflagellates, which have large amounts of saxitoxin in them, the concentration will accumulate within these filter feeders and fish. So they will have a high concentration of saxitoxin within them. As humans consume these organisms in that food chain, now humans will end up being affected by this toxin. Saxitoxin works by binding and blocking voltage-gated sodium channels, and this prevents the passage of nerve impulses and can lead to respiratory paralysis. In review, today we have learned a brief introduction into ion channels and nerve transmission. the cell membrane charge imbalance, voltage-gated ion channel overview, voltage-gated sodium channels, voltage-gated potassium channels, voltage-gated calcium channels, gated ion channel responsibilities, neuronal action potential explained, the propagation that carries the action potential, and the actual cease of neurotransmission. We also discussed some examples of toxin effects made by certain organisms on ion channels and how they affect humans and other organisms at that. Included are my references. I hope you enjoyed my presentation. Thank you very much for your time.